Okay.
It was my wife.
Okay. Okay.
Okay. Third, but not least, Francesco, that is going to speak about gene networks and geometric deep learning. Thank you, Francesco.
Thank you very much, Maurizio, for the invitation, and to Rita for organizing this beautiful meeting and financing everything. So let's start. I usually work on machine learning and deep learning projects with a group of people that share passion and interests, and we call ourselves Curious AI Lab. So I've listed the most senior one. So Maurizio is the leader. We gather around him and choose what to do, curiosity driven. So this is work in progress research that used idea from many people. Some disclaimers before I start. This is super work in progress, and there is no publication yet. The results need more experiment to confirm what we found, but still, I think it's interesting, and I wanted to speak of something that is really geometric deep learning and how to use geometric graph neural networks. And second disclaimer, there is some biology. I am not a biologist, but I will have to explain to you a little bit of biology, because otherwise it is impossible to understand the peculiarities of the data sets we are talking about that needs us to invent some new technique. And finally, this is very applied research. There were so many awesome talks before and yesterday, the days before, with theorems, with insight, with a vision. This is applied research. I won't give proofs of stuff, but there is mathematics inside everything after the biology. So we start with biology. The task. The task is this is a strange word. Single cell RNA sequencing. It's a protocol. I will talk about this. And we want to go from this kind of data set to classify cell types. Okay, so the task is something that's called like this and is classifying cell types. So I need to talk a little bit about cells and about RNA. Maybe many of you are familiar with this, but I think it's interesting anyway. Of course, you know that all the cells inside every organism, inside your body, inside a cat, inside a plant, they all have the same DNA. The DNA is the same for all the cells in an organism, with some exception, with some mutation, maybe with some cancer is different and everything, but still we have the same DNA for all cells. But cells are very different. If you look to a large organism, you find many kinds of cells, and they all have the same DNA. They work using the same DNA, but they are so different. We will focus not on so many different cells, but on immune cells, T cell, B cells, and so on. They are less different, but still very much different. Okay. It was the touch. The way they are different is that they don't use all the DNA. They use some part of the DNA, some specific part. You know, RNA is what brings information from the DNA to build proteins and do other stuff, regulatory and everything. So you must think of your DNA as something that is usually zipped and closed and inaccessible, but then specific genes, specific zones of the DNA are made accessible, and then the RNA polymerase goes and copies these pieces of DNA in RNA. And these molecules that go free inside the cell, they start in the nucleus, then they go outside the nucleus into cytoplasm. They can be found there, and they are what actually the cell is doing. So the cell is not using all the DNA. The cell is using some specific genes. And that's how different cell types can use the same DNA. They use specific parts of the DNA, not all of that. And this complicated named technique, this protocol, single cell RNA sequencing, is a technique that allows you to take a sample of tissue, and then from every cell inside the tissue, go and count the number of molecules of RNA of each gene. So you detect the molecules and count them for each cell, and each gene in the DNA, in the library, you can get a count. How many molecules of RNA of this gene are in this cell? So the task is easily explained. You have, for a cell, the counts of all genes. Some of these will be 0 because the cell is not using those genes. Some will be not 0. And then you have to identify this cell is a B-type cell, this cell is a macrophage, I don't know. Okay, seems easy. So, and single cell RNA sequencing is not a minor technique. It's becoming cheaper and cheaper, and so it's becoming customer in clinical studies to spend some money and have also this kind of analysis performed. And then the results must be studied in some way. So there are software, there are protocols, techniques. So we are trying to give a contribution to something that is important for the biology research and for medicine. So this is what a data set looks like. You see the rows are cells. There is a complicated reason why they are identified with the nucleotides hashes. They are random hashes and here in alphabetical order, but the rows are cells and the columns are genes. The genes are named linked to the function or something like that. And the last column is the label, the class, because we are doing supervised learning. In a moment, I will tell you. So for each cell, we know which class it belongs to, which kind of cell it is. So we can then do some supervised learning to try to take this row of counts and decide what kind of cell it is. So there are many common approaches to this kind of data sets because, as I said before, they are very important. And the most common approach doesn't use neural networks, and it's simply, you usually don't have the labels. Okay, the labels, we will see how we get them, but you usually don't have the labels like in this data set. So you get only the counts, and in some way you do some clustering. You try to find which cells are similar to others, which rows are similar to others, and then maybe with a biologist you try to understand each cluster, what kind is it, and these are the two main software that are used. But there is some work also on neural networks. Another important package is SCVI that does variational autoencoders on the counts, on the cells. And then the clustering is performed in the latent space. And then there is the other alternative that is quite common. It is to still not using neural network in this case, do self-supervised learning with pseudo-labels. What does it mean? It means that you do, once you do clustering, you do unsupervised learning, and then you assign labels to some huge data set, to some atlas, and then you train something simple. Usually they use just logistic regression that, of course, can allow you from a vector of genes to classify the cells. In this case, the types. We call the labels pseudo-labels because they are not a real ground truth. We don't know if the labels are perfectly correct. We found them from some clustering. But still, why use only logistic regression? So our intention is to try to improve on alternative tools. So we will be doing self-supervised learning using pseudo-labels, but we will try to use a neural network instead of logistic regression. What are the problems using a neural network? Okay. I need to talk a little bit about the challenges, about the problems of these data sets, because it's, as of now, I've hidden all of them. So, first of all, the protocol has extremely low efficiency. Inside the cells, there are these RNA molecules, and then at the end, you get counts in the row, but you don't get a one-to-one mapping. Not for every molecule you get a count. You lose some. How many? I don't know, 97%, something like that, okay? You lose, it depends on how many cells you are analyzing at the same time. If you analyze a smaller number of cells, maybe your efficiency goes to 70% and you, to 30%, and you lose just 70% of the molecules. But otherwise, if you're analyzing a lot of cells, then the efficiency go down. So you don't see all the molecules. And many genes, in fact, are not very much expressed inside the cells. There are genes that are maybe important for the cell, but they are very critical. And so if in the cell there are too many molecules of that gene, then maybe the cell doesn't work correctly. And so if you have low efficiency and some important genes have low expression, then you often get zeros. You often get zeros where you should have 1 or 2, maybe. And in the end, these matrices are sparse. Most counts are 0, but of course there are genes that have a higher average and so on. And as I said before, many zeros would be non-zero with a higher efficiency. Okay, another problem is the high dimensionality. The number of genes that I call capital N is of the order of 15,000. And then the last problem is that the data are not continuous. You get small integers, zeros, ones, twos, 19. And okay, usually people want to use Gaussian statistical techniques that rely on the Gaussian assumption, and this is no good. So what usually people does, and we will do it also, but I encourage any of you who has understanding of probability theory to try to get something better, because I think this is one of the major points that we're missing. What people usually is to transform the counts in expression levels. So I will call the row counts, the integer numbers R, and you have the counts for cell I, for gene J, and then you transform these to transform this to xij, the so-called expression levels, and the standard transformation is this one. So basically, okay, Ri star is the average of the cell of the counts. So you, in some sense, normalize with these coefficients the counts with a scalar coefficient in such a way that the sum of the counts of the row is 10,000, arbitrarily decided by someone sometime. Then you add a pseudo count of 1 because you're going to compute the logarithm and you have many zero counts. And then this is the expression level. So this is how it is usually done. Okay. Other challenges. You must understand that genes, which are the features, the columns of the data set, are not all equal. There are several families with different behavior. There are genes that are needed for the proper functioning of all cells or almost all cells, and you expect to find those genes expressed in all or almost all cells. So these won't be very useful to classify the cell type if all cell types use them. Then on the other side of the spectrum, there are genes that are specific for a cell type, marker genes. And some of these are known to be marker for one type, for two types, for a family of types. Some are known to be constitutive. Many are not known to be either, some in between. Another thing that one must understand is that there are many genes that don't really, okay, maybe they code for proteins, but that's not they are not building stuff, walls and tools and furniture. They are used to regulate the expression of other trees or genes. So, as I said before, these kind of genes are very important and the cell is sensitive to their quantity. So there are genes that are really important at a very low expression. And you don't see them very often. Okay, and last thing, that since the genes are so different and we don't have a library which says every gene what does and how you can use it, and you have 15,000, usually what you do, you rank genes by some kind of variability in order to have the constitutive genes with low variability on the tail of your list and marker genes at the head. And most methods, this is a major point, this is very important for what I'm going to explain to you. Most methods restrict themselves to maybe 2,000 or 1,000 or 500 highly variable genes. So usually you have 15,000 columns in your data set. The first thing you do, you drop 80%, 90% of the features and concentrate only on a few thousand. And this, I think, is not very good because you're losing too much. You need to do some kind of reduction, but not this way. Another difficulty is that cell types are not 7 or 12. There is a continuum of types, and they are in a hierarchical structure. And if cells of CD34 plus can become lymphoid and myeloid, you will find all of them, the lymphoid cells, the myeloid cells, and the progenitor cell that has not yet decided what to become. And this is the same for all the points in the tree. And this is subtree of the immune cells, not of all the cells, okay? So you have all kind of, they are not like classification of images in neat categories. And these labels that we are using are assigned with some kind of clustering in some kind of projection into dimension or something like that with the various algorithms. The biologist takes the data sets and do some projections, some clustering, then tries to understand in each group of cells which marker genes he can see, and then assigns the types. And so we get our labels. So to sum it up, we have expression labels used as input. Then we have pseudo labels used as output. And we want to find some neural network that acts as a classifier in this setting. And I must say, there is no foundation model for this kind of problem. So for images, we know there are convolutional neural networks. For text, there are transformers and everything. For single cell, you don't have anything. So your basic idea is to use a multi-layer perceptron. That is really not very good still. So we investigate architectures. And the problem in which we are trying to find the best architecture is not a very important problem. So classifying with pseudo labels is not so important. It's just something that allows us to test different architectures in a fast way and get an accuracy back. And so if you find an architecture that works well with this kind of data, then maybe you can use it for a variational autoencoder or for something more sophisticated that doesn't necessarily use pseudo labels. So we are looking for the architecture that is foundation. So some words on the actual data set where the experiments were performed is a 10,000 cells PBMC. It's maybe it's written peripheral blood mononuclear cells. 10x is a company that does this kind of analysis. So immune cells from the blood of the Homo sapiens, and 10,000 cells more or less, and 50,000 genes more or less, 13,000 genes. So M is the number of cells, capital N is the number of genes. There will be small n and capital N. So capital N is the total number of genes, 13,000. It is very sparse, 89% of zeros, and okay, you can add the numbers and see how sparse it is. And if you focus on a gene J, and on that column, you have many zeros and some non-zeros, and you compute the average, you would say, okay, there will be genes with high numbers that are often found. Well, 80% of genes has average count less than 1/5, and 15% has average less than 1/100th. And 5% of genes have counts only binary, 0 and 1. This is also a problem from a statistical point of view, because if for a feature you have only 0 and 1s, you cannot compute two statistics. You can compute at most one. You have only 1 degree of freedom. And this is what it looks like. I've taken just a bunch of genes which are not at the beginning and not at the tail. So these are common genes. You see, they are not all zeros. They are not all zeros and ones, but something like that. You compute the expression levels. These are the expression levels. So the zeros stay 0, and then the other numbers usually grow, and how much depend on the cell, because they are scaled on the average of the cell. And so, for example, you see that this one and this one become, okay, no, they become the same thing. This is really strange. Okay, I will have to check this because it doesn't. Kind of strange this. Okay, and we have 85 classes. And this is not a strange number because common software often have 100 types and subtypes if you go really to a small mesh in the cell types because of the continuity of groups I was saying before. These classes were found with a statistical software that ensures that they are statistically uniform genome-wide. So looking to all the genes, they are compact at least. But we didn't have the time yet to do the biological analysis and say each one in what point of the tree falls and what kind of cell it is. And these 86 classes, they are not balanced, of course. The smallest one has 17 cells, just 17, and the largest one 400. And as I said before, genes are not all genes are equally important. So we sorted the genes by variability. So the first genes are most important and the last one the least important. And I recall we will be using a variable number of genes, 100, 1,000, 5,000, but usually other people use 2,000, typical. So vanilla models. The first thing you try is what usually people does, that is logistic regression. But before, I was forgetting, there is one slide before logistic regression. So in all of the experiments that I will be talking about, we take small n genes, and small n is variable. We try with just 100 genes, the most variable, and then we grow until capital N using all genes. We use Adam W optimizer. We use a strong L2 regularization, standard learning rate. Why strong regularization? Because we will have a lot of parameters for the number of cells, and so you have to regularize to get something better. And we replicate all important experiment at least 3 times. We do a standard split in training, validation, and test, and the split is stratified for labels. And we measure the test accuracy for the model that we, in each experiment, while we train, we select the model which has the highest validation accuracy, and then we measure the test accuracy for that model. Okay, logistic regression. In logistic regression, you have small n inputs because we are using from 100 to 3,000, 13,000 genes, and you have are immediately connected to the classes. So I put here 8 classes. The classes are 86. I put here, I think, 40 genes. The genes are at least 100, maybe 13,000. And so the number of parameters is the product, and so goes from 9,000 to 1 million parameters. And in this plot, all the plots will be like this. You have the number of genes used as input for x-axis, and then the accuracy, the test accuracies in the y-axis. And you see that if you use a small number of genes, you don't have accuracy, but then it goes up steadily, and it is maximum at more or less 2,000 genes, which is what usually people use. And I must say, the pseudo labels were the clusters that decided the pseudo labels were computed using software that used the first 2,000 genes. So maybe this is a little bit fishy. It's using the same genes, and so this is the best. Of course, this is the best. And then it goes down. It goes down quickly. Of course, only validation and test accuracy go down. The train accuracy is 100% almost immediately when small n is large. because you have too many parameters. You have just 7,000 cells in the training set, and you have 1 million parameters. You can regularize as much as you want, but you go in heavy overfitting, and so you have to use a small number of genes. Then you try an MLP. Okay. You try an MLP. We've tried with more than one hidden layer, but the best results are with one hidden layer. So we put a hidden layer with 64 units, which is in the order of magnitude of the outputs, and the number of parameters is a little bit less because almost all parameters are in the first layer when you have small n times 64, instead then small n times 86, and then a few thousand parameters for the second layer. And the performance is a little bit better. No, it's quite better. It's 10% better, more or less. No, 5%, no, 10% better. And this is MLP, and this is logistic regression for comparison. And still you have the best result for around 2,000 genes, and still you have a decrease when n goes up, and you lose everything. You see, you go back to the values of the logistic regression. Okay, so we want to move forward from these vanilla models, and we want to use graph neural networks. Why graph neural networks? Because what you really want to improve the models is to have some sharing of the weights. So you don't have to use all those weights that are needed for the MLP. Maybe you use a small number of genes in the output, but in the end, but you integrate information between genes, possibly using gene similarities. And there is such thing as graphs of genes. These are called gene networks. Gene networks is an argument that is popular, and there are there are a lot of publications that refer to gene networks, and so there's a lot of stuff here. How do these gene networks work? There are many different ways to compute a gene network, and many are hand-tailored. Many are just not genome-wide, but just for a bunch of genes. What we want to do is have a quick graph, a quick network that is genome-wide, and usually in this case you take a co-expression matrix, correlation. You see which genes are expressed together to other genes, and you connect this one. So you start maybe from a correlation matrix. I've taken here a bunch of genes. This is a square symmetric correlation matrix. I put zeros on the diagonal just because otherwise the heat map would colors couldn't be seen. You see you have positive correlation, negative correlation. They seem small, but they are significant actually, most of them, not all of them, and statistically significant. And so you have the correlation matrix, and then you apply a hard threshold to get an adjacency matrix. For example, filter all the cells with value greater than, I don't know, 0.009, and you get this, which is of course a symmetric adjacency matrix that links your genes in some way. So these genes are co-expressed, they work together, they are found at the same time in the same cells often, more often. Okay, you don't have a correlation 0.9, so not so very often, but still they work together. Then you can take several graphs, actually. You can use other thresholds, maybe a higher threshold to have a subgraph, a lower threshold to have a larger graph, or maybe you do the opposite. Now you threshold the values which are negative below some other threshold, and you get a completely different graph that says which genes work against each other or are not found very commonly together. So we have all this richness, and we want to use these graphs. Okay, the coex matrix again is from Cotan, and we for the final model we use two graphs, thresholding I wrote here. And okay, so we want to use graph neural networks. How we do that? First of all, this is not the typical GNN task. Why not? Because you don't have many different graphs or many different zones of the same huge graph. You have this gene network graph or graphs that are fixed. All your samples have the same graph. What changes from a cell to another? The sample is the cell has to be classified. The cell has expression levels for each gene. So in each node, nodes are genes, are the features. In each gene, there is the expression level. And you have the whole graph with all the expression levels. Many are zeros, some are not. And you have to classify these features to a cell type. And you have only, in each node, you don't have many features like often happens with graph neural networks. You just have one number, one scalar number, which is the expression level. And the task is graph level. You have to classify the whole graph. You don't have to classify the nodes. You have to classify the graph. And moreover, and this is the most difficult part, nodes are not really exchangeable. You're not really, you're not going to be able to modify the graph as usually can be done in other problems where equivariance is true and complete. Here, if you change to the counts, the expression levels in two regions of the graph that are really, really similar, you get to completely different cell types because marker genes are specific for cell types. You cannot mix genes with other genes. They are not the same. Okay. Since nodes are not exchangeable, global pooling does not work. Usually when you do graph level task, you do some layers of graph neural network, and then you end with a global pooling of some kind, and then you classify after the global pooling. But the global pooling loses all information of which nodes brought those features, and you cannot do that. So we need a different head. So the basic structure we came up with that is shared with all the experiments, even the last one, which really works, is the following. So this first disk here is the input layer. So you have values inside these. The nodes are the genes. They are connected by the graph, and you go down to the first graph layer. I don't know. Okay, so you start with all genes. The white are the all genes, capital N, 13,000. Then two graph neural network layers. The first one, you see, these have a depth, they are high because the first one, you start with scalar values, you compute 16 features. And then the second one, you go back to one feature. Why? Because I cannot do global pooling. And so I have to do an MLP to end. So how do I do an MLP? I choose a subset, I restrict to small n nodes again. So I have the two graph neural network layers, use all genes and integrate information of all genes. And just only after that, I drop most genes and use only 100, 1000, 5000 genes. And then in the end, there is the same MLP as before with a hidden layer with 64 units. Maybe one can find a better architecture, but still this allowed for easy comparison, because basically you're using the same genes with the same final architecture, but you have some extra information that is integrated by the graph neural network. And then one can add several ideas, do a residual connection, residual branch. Then you can, okay, you don't like one-dimensional input, you can add features that are of that gene, not of that cell. You can add, okay, this gene is particular. Maybe this gene, I know it's constitutive, or maybe this gene has a high average read counts. And so you can put other features which doesn't change with the cell, and give some extra information. And you can use several graphs in parallel. As I said before, you can use several graphs for the first layer, then you concatenate their output before the second layer. And so there are all these kind of variants that we tried. And we started trying with the common GNN layers. The most used, apart from maybe GAT, is GraphSage. GraphSage is a simple architecture in which when you compute a node I in the subsequent layer, you have the features of the same node with a matrix, and the features of the neighbor nodes averaged in the neighborhood, and then multiplied by by a matrix. So computes the average in the neighborhood, and then the features of the node, and do some parametric combination of these. Very easy. GCN, same year. Okay, it's quite similar. There is, you don't sum over separately xi and the neighborhood, you sum over the neighborhood and xi, and you apply, so you apply the same transformation, the same parameters, and the difference is that while this computes the average with this is the arithmetic average, okay? Every neighbor has weight 1/n, the same. This computes a weighted average that weights less nodes that are high degree. This is similar to the degree, so similar to the cardinality of n and j, and I and nj. So here you have some kind of weights in this average. And then there is attention. There are at least two architectures. I used here the version 2, which is probably more fine, in the sense that it has better performances. And this is really, really more similar to what we want to do. Because again, it's an average. These alpha ij sum up to 1 if you sum them over j. So it's an average on the neighborhood, but the average is intelligent, or at least it tries to be intelligent, because the weights comes from the attention. So the weights depend on a trainable vector AT, A transpose A, and the output, the tentative output, you see W2, which is the matrix that applies to the average of the xj, is applied to the xj. So you try to see what happens if you apply W to xj and another W maybe to xi, and see if I get some kind of vector after a nonlinear function, and then I weight this vector in some way, and this gives me the weights. So I know I can learn which neighbor I have to weigh more. Sometimes it works, but sometimes it doesn't. Nothing of this works, nothing. The best performance of all the experiment is worse than the MLP. Here I've put a disorder subset of experiments. You see the MLP is in yellow here, and the maximum has never been surpassed. There are some models that looks promising in some sense. Experiments are quite long and heavy. Some models don't really converge, and so you get maybe 2% accuracy, which is a random classification. So nothing works. And the problem, when you think about it, is that you are trying to average the neighbors in an intelligent way, or maybe not intelligent if you use SAGE, but you're not doing it well enough. In gene networks, you have a gene network, you have neighbors. What are these neighbors? What characterize them? They have a correlation, co-expression. They are correlated to the central node, but they are not necessarily similar to each other. They have small correlation, and they may belong to different families of genes, and they may be correlated for different reasons. Maybe one gene is correlated with another 100, and of these 20 share one kind of cell with this gene, and other 30 share one other kind of cell with this gene. So you must try to sum over different families and not sum over everything. And the attention doesn't work, doesn't learn the weights as it should. And so we have to perform these averages. And so we engineered a new architecture that does so. And okay, we also tried multi-head attention, because we understood that we needed more than one average, because the attention with a number of heads equal to 1 computes just one average, but you have to compute several averages inside the neighbor because you have several families of genes. So multi-head GAT still doesn't work. And maybe the point is this: if you want attention to learn how genes should really be connected for your task, you must provide enough information of these genes. Not just the read counts, the expression level, maybe the average of the gene. You need to have a long vector of features for nodes if you want attention to work. And you don't have that yet. So we, tentative name, tried with this cheat attention network. So it's an attention network that it's not self-attention. It doesn't reflect on itself, on its output. It just peeps at another network and try to copy something from their paper. Okay, so that's the tentative name is cheat attention. Okay, we add as input features, which will be treated differently from the expression levels, specific signatures of each gene. So each gene will have a signature of itself. These signatures are vectors. We use dimension 64. And we want to train these signatures. We don't have them. So we want to train them in such a way that genes that are similar for our task will have similar signatures. How to do that? We go back to our MLP and we cheat copying from it. So consider the basic vanilla MLP, but the MLP that uses all genes, because we need the signature for all genes. So this is the last model with n small n equal to capital N of the MLP experiment. And you have the first layer with weights W0 that goes from R capital N to RD, which is the number of hidden units that was 64. And then the second layer that goes from 64 units to 86 classes. This matrix of weights, when it is trained, has n columns, each one of length 64. So we have a trained signature for each gene. They are not the output of this network. They are the weights of this network. So I want to build now a graph neural network that uses as input features the values inside the weights of another network. So it's looking really, it's peeping inside the other network. It's not just using its results, it's looking into its brain. And the signature are the weights. Okay. There are other differences. These signatures are used only for the weights, for computing the alpha, only for the attention mechanism. So these are the alpha of our cheat attention network. You take the weights, the signatures, you take the signature of gene I, and then you, okay, this is an error, should be capital K. This matrix, capital K was the number of head of a multi-head GAT. So we don't have a vector here, we have a matrix because we are multi-head. So this signature is multiplied by several vectors to obtain the same number of coefficients alpha. And then, okay, so the attention is not depending on I and J, but just on I. We use multi-head attention, as I was saying before, this D prime should be K with 64 heads. And this is the formula of the attention of the layer of the graph neural network. So you must use this together with this and with this. This is similar to the multi-head GAT that was written before. And in fact, we can make a comparison. Our cheat attention is like this, while the usual self-attention is like this. This first operator is just the concatenation of the heads. Then, okay, we have different W for the central node and the neighbor node, but that's not a big deal. And then, as we see, we have this alpha that depend on I and not on J. Okay, in this case, depends on J, not on I. Maybe better to say it like that, and depends only on the signatures. So what are really the huge differences here? The first thing is that we don't ask the attention mechanism to learn these weights from nothing, from the output. We learn them from the signatures because we are cheating. And in this way, we can have a large value of K. Either you have a large value of K or you have a large dimension of Wx. Why? Because when you're computing Wx and then concatenating on K, you end up with a number of features that is the product of K times the output dimension of W. So if you want K large, you need to have W with small output. But W, the output of W is used in the self-attention to compute these weights. So you want a high number here, and in GATT you need to use a small number here. That's arithmetics. Instead, we don't use X in computing these weights, this alpha. We're not using X. We can use the signatures that we want of the dimension that we want, because they are completely separate from the dimension of W. Here, X for us has dimension 1, because I'm not using the signature as input feature also in the layer. I'm using the signatures only for alpha. Here I have a scalar, which is the expression level. And so I can get a large value of K. So, and GATT and GCAT works. The purple line is what we get. You see, is a little bit better in the best, for the best N. And okay, this blue was the maximum over all the other experiment with graph neural networks. So you really must compare the purple with the blue. All other architectures perform like that. We've tried everything. And it is so better than use the other graph neural networks for this task that is even better than the MLP, which was incredibly good in the end. And moreover, it is stable. It doesn't go down so fast as before. It's stable at around 60%. And for the architecture, but I finished my time, so it's not important. We used 2 graphs, and the first layer was cheat, and the second layer on a standard convolutional. The number of parameters is more or less double of the MLP. And okay, the maximum TA is 65%. And that's all.
Very much, Francesco. And questions?
So I'm also interested in the RNA expression, but so we use it to classify some reaction of the body to pain, no pain, this type of questions like that can be hidden in DNA.
Mm-hmm.
So have you looked like at looking at the DNA to predict some body reaction, some illness, some type of things? You see what I mean, right?
DNA or RNA?
Sorry, RNA, transcriptome, of course.
Okay, no.
RNA, I meant.
Here we are looking for a foundational model. So we used a data set of a healthy patient, and so it's everything is stable. But after, when you have such a model, then you can try to apply it to usually. Single-cell RNA-seq used for study of cancer, of pathologies, so maybe inflammation.
You took it from ChIP-seq?
Yes.
Okay, but what was the technology?
10x. 10x.
10x, okay.
10x.
We use Illumina.
I think it's the same.
I was going because I don't know
the
that.
The company is 10x. I think
Illumina
is the protocol, I think.
Perfect.
I'm not sure.
Okay, so we stay in contact. When you reach the perfect, then you come to us, that we have the data.
Thank you, Rita. Other questions?
Yes, thank you very much for your talk. Maybe an outdated question, but did you check how well random forests perform on this kind of data? Just
how well?
Random forests, like decision trees, and then
yes, they perform worse than logistic regression.
Okay, thank you very much.
A little bit lost. Huh? Okay. Depends on the task, classify and so on.
Yes, random forests are always very good. We know. Other questions? If no, let's thank the speaker again. Okay, we meet at 2 here, but first don't move our powerful chair as a very important announcement.
So together with the organizer, this is especially to the people that are here that have written a poster or maybe have not written a poster but wish to have written a poster.